So, good morning, class. Today we are going to see the very first and fundamental thing about the whole biology subject, and this is about the cells. So, today in the subject Zoology 2 and Animal Physiology, we are going to talk about the cells, or we can say the building block of any living organism. So today we have to see that what are basic kind of the cells which have been present in the entire living organism. So these are the two very fundamental type of the cell which basically build the entire living organism. You can say their organism system can be just based on these two kind of cellular organelles, prokaryotes and eukaryotes. Now, these are the following aim and objective which we needed to be fulfilled during our lecture. We have to study the cell, prokaryote cell and eukaryote cell. We have to see what are the key differences between a prokaryote cell and the eukaryote cell. We also have to see what kind of the similarity are obtained in both the cells, or you can say the common features is present also in the prokaryote cell and eukaryote cell. So, by following these objectives, so we will be get a total knowledge about the prokaryote and eukaryotes. Let's begin with the diagram. So, this is the diagram of a prokaryotic cell and eukaryotic cell. You can initially see the difference. Okay, the prokaryote cell, they look just very simple. Okay, there is no very much complexity you can see inside a prokaryote cell. On the other hand, if we see the eukaryote cell, they are showing the multiple organelle inside them. The very complexly present every cell organ inside them. Their cell is little enlarged. So these are the two fundamental differences, or you can say just by seeing them, you can have an idea that these cells are a way more differences in their structural organization. Let's begin with this. So basically, this whole self-evolvement, or you can say the architecture of all the organisms that are basically, we can say that these are the two remarkable things. Like we can call the particular organism that is a prokaryote style, and we can also come some other organelle by the eukaryote style. So basically, when we are denoting this kind of pronouns like the prokaryote style or eukaryote style, so basically in that case we are just deriving the what kind of cell system is present in that particular organ. Let's start with some characteristics. So basically, if we take their ontogeny, the origin on the earth. So prokaryote cells were here first, and for billions of years were the only form of the life form. So these prokaryote cells, they might be simple, but they are the old one. They are present on a very first sight of the earth when life began on the earth. So they are the ancient one, while eukaryotes, they are the modern cells. You can say they come after a way more time after the prokaryote cell on earth. So all the prokaryote organism, they are unicellular. This is their key feature that all the prokaryotic organisms are unicellular. So eukaryotic cell basically appeared on Earth as after long, but they are the most advanced one. Maybe they come later or they come after the prokaryote cell. But if we talk about their advanced features, then eukaryote cells are way more advanced than the prokaryote cell. As you can see that these prokaryote cells, they are only unicellular, while other hand these prokaryotic cells, or other hand on these eukaryotic cells, they are basically they can be unicellular or they can be multicellular. So despite all these differences, there are also some things which in common also, but there are huge difference in between the prokaryote and eukaryote cells. So let's begin with the characteristic feature of the prokaryote cell. So firstly, we will see how the prokaryote cell having some characteristics. So first characteristic of prokaryote, that they are the simplest type of the cell. So any simple cell which have some basic characteristic, all the basic characteristics should be present in the prokaryote cell. So these are the very simple kind of cells. Oldest type of the cell appeared about 4 billion ago. We can call them, these are the ancestral cells. They are the most old cells present on the earth. As we already discussed in last slide, that the eukaryotes are the new one and the prokaryotes are the oldest one. They are having the largest group of organisms. So if we talk about the group of organism, a number of the organisms which are having the prokaryote cell, so they are having a large variety of organisms which have their cell structure is a prokaryotes. They having the unicellular organism and found in all the kind of environment. So, further on their characteristic, they are the unicellular organism, and they are, you can say, they are abundantly present in the every or each kind of environment. They can be present in the hot springs. They can be present in the humid one. They can be present in the desert. They can be present in the place where rain is common. They can be present in normal circumstances. So every circumstances they are present universally everywhere. They do not have a nuclear membrane. This is also a very important character when we study also the differences between prokaryote and eukaryote. So this is a major difference we count in between them. They are having a nuclear membrane. So this is a major difference between a prokaryote cell and eukaryote cell. That prokaryote cell do not have any nuclear membrane. Their circular shaped genetic material dispersed throughout the cytoplasm. So basically, the genetic material, the DNA, the chromosome, they are having the circular shape of genetic material. Commonly, in eukaryote cell, the genetic material is linear in shape, but in case of the prokaryotic cell, the genetic material, the chromosomes, are basically dispersed through the cytoplasm, and their shape will be the circular shape. Please remind this that always the prokaryote cell, they are having the circular shape of genetic material. Prokaryotes do not have membrane-bound organelle. So, as their nucleus is not bound with the membrane, so though there are all other organelles which are present inside a cell, they do not have any kind of membrane inside them. So this is also very important feature. Whenever we are talking about the prokaryotes, this is you have to be seriously taken that all the organelles inside a cell, they do not have any kind of membrane. So prokaryote have a very simple internal structure, and if we talk about their size is smaller than the in comparison to the eukaryote cell. Now you can see some shapes of the prokaryote cells. They are very small in shape. Here you can see this is a spherical shape. Okay, so spherical shape is like a helical and spiral. This is the bacillus rod shape. You can say these are the little elongated one, but not very thick. They are the thin elongated rod shaped bacteria, bacillus. And also you can see the spiral. So spiral is like the spiral binding you see on the notebooks. So same the helical spiral binding structure can be seen. Cocci, this cocci is basically the round and oval shape. So these are the steps which are very abundantly found inside the prokaryotes. The round and oval shape, the long, elongated and thin shape, the spiral shape. Mostly these are the common shapes for the bacteria. Bacteria also have these kind of common cells in them. So these are very common shape for the prokaryote cell. This is the structure of a simple kind of prokaryote cell. You can see this is the outer membrane. Okay, whole cell have to be covered by a membrane that is called the outer membrane. Or if we are talking about a plant cell, okay, if we are talking about the plant prokaryote cell, so they will be having a cell wall outer them. Okay, cell wall have to be covered the entire cell membrane. After that, you can see the capsule present. This capsule situation that is not present every time, but in certain situations, the capsule can be present or can be not. That basically depend on what kind of bacterial or prokaryote cell it is. Maybe if sometimes cell have to be present in a zone where the complications are more, at that time capsule have to be very rigid. The third thing which come through the inside, this red one you can see here, this is the plasma membrane. Okay, plasma membrane is the first or membrane which cover the plasma, the cytoplasm inside the cell. This is covered by the plasma membrane. This is the dispersed nucleic region. Okay, there is no definite shape of the nucleus, which is like in the eukaryote cell. Their particular membrane bound nucleus is not present in the prokaryote cell. So the nucleotide region, this is basically just dispersed throughout the cell. And then the ribosomes. So ribosomes, you can say, along with the nucleotide region, there will be the ribosomes. Ribosomes have to be needed to be present into the your prokaryotes. Why? Why the ribosomes are here? So basically, all these ribosomes, they are the protein factories. So nucleotide might get the code for the ribosome to make special kind of protein. And if there is special protein formation will be take place, then only the cell perform their function. That's why the presence of ribosomes is very important. Six is the outermost, you can say, these hairy growth-like structures. These are called the palace. Okay, these are the basically hair-like outgrowth which help the cell to attach with the substratum, and sometimes it is also help in the protection of the cell. This one, the last very long thread-like structure, they are called the flagella. And flagella basically help the cell put into their locomotion property, into movement. If the cell wanted to do a movement, then the flagella is the thing which basically help the cell to the move from one place to another place. So this is all about the prokaryote cell. These are all the features which present inside a prokaryote cell. There is no other complexity, no other major like ER bodies, cytoplast or lysosomes, mitochondria. These organelles, the complex organelle cell organelles, they do not present inside the prokaryote cells. Now we will see the characteristic of the eukaryotes. So if we compare eukaryote and the prokaryotes, they appeared approximately 1 billion year ago under their ontogeny or origin. So they're not as old as the prokaryote cell. They are the newly developed cell, just have the origin 1 billion years ago. Eukaryotes are generally more advanced than prokaryote, but if we compare their advancement, these eukaryote cell, they are much more advanced in comparison to the prokaryote cell. Nuclear membrane mostly surround the linear genetic material DNA. So this, like we already studied in prokaryote cell, their genetic material is just spread throughout the cell cytoplasm. But in the case of eukaryote, their genetic material is surrounded by a membrane. So these presence of this membrane in the nucleus and the other organelles of the cell, these are the key features of a eukaryote cell. Next characteristic, if we see in the case of eukaryotes, they have several different parts. As not in the prokaryote, they do not have major categories under the cell organelle, but in eukaryote, they have the several different parts. Like mostly the prokaryote organelle have covering known as membrane, but in the eukaryote structure, they have the complex internal structure. If we compare a little outer coverings of prokaryote and outer covering of eukaryote, then you can say that the prokaryote covering is slight complex, but eukaryote internal structure is more complex because The cell, the prokaryote cell, they are little vulnerable cells. They have very simple thing. They do not have a very defense mechanism to work with outer dangers. So basically, they need to protect their outer covering. They have a like precautions are built into. So they take the precautions, okay, but you because they do not they know that they do not have a very good self defense mechanism to fight against any kind of disease or any kind of harmful thing. But eukaryote cell have a complex defensive internal structure which is very efficient to perform any kind of function inside the cell. So eukaryotes are larger than prokaryotes in size. If we talk about their size, so eukaryotes are way more larger than the prokaryotes. But it also depends or vary from species to species. You can see here the structure. These are the structure. So whatever the name here you are seeing on the slide, they are not present inside the prokaryote cell, like mitochondria, Golgi complex, nucleus, bounded nucleus, endoplasmic reticulum, and this cell membrane. So these characters they are not present inside the normal prokaryote cell, or if normally prokaryote cell do not have any organelle which have the membrane bound thing. So this whole membrane bound concept, like the mitochondria, they have a membrane, the Golgi, they have membrane, ERs, they are having a membrane, the cytoplasm, we will take it later. These all things, basically, they're characterized by having the membrane in them. That's why they are come under the eukaryote cell. Now, if we see some major differences in between a prokaryote cell and eukaryote cell, so you can see here in prokaryote, organs lack a membrane. Okay, they do not have any kind of membrane in them, while eukaryotes they have all the members covered by a membrane. Prokaryotes, ribosome are the only organelles. So if we see through clearly, they're just having one special cell organelle that is ribosome, or that is why it is present. I already told you that they are the protein factories, and if there are no protein factory, it will be happen inside the cell, then there will be no functional working can be done. Eukaryote, multiple organelles including ribosomes. So they are having ribosomes. With ribosomes, eukaryote also have different kind of cell organelles present inside the cell. Genetic material floats in cytoplasm. So in the case of protoplasm, they having genetic material but this is spread out through the cytoplasm. DNA and RNA both just they are floating inside the cytoplasm. They do not have a definite place to be in. But in the case of the eukaryotes, membrane covered genetic material. So they are having a well covered nucleus inside their cell, which basically covered the whole genetic material inside the cell. Circular DNA, in the case of prokaryote cell, there will be a circular DNA, but in the case of the eukaryote cell, there is a linear DNA. Cells are smaller in size, so the size of the cell, if we compare, their size is small. Eukaryotes have a large size. Has large number of organism, they are having the small number of organisms. Small number of organism, but they are the complex one. Appeared 4 billion years ago, there appeared just 1 billion ago. So this is a summarized version of whatever you studied till now. These differences are basically based on them. Now we are done with the differences. Also now we have to see what kind of similarities present in between two cells. So first here we can see the diagram also. The both having the cell membrane. Okay, the outer covering of the cell you can see here. They are also having the plasma membrane, and also this eukaryote cell, they are also having the plasma membrane. Both type of the cell have ribosomes. They are also having ribosomes. The eukaryote cell also having the ribosome. Both the type of cell have DNA, means both the cells are carrying the genetic material, because without genetic material the cell cannot live, because cell have to be work on some instruction, and these instruction come from the DNA unit. So that's why both the cells, they are having the DNA as a genetic material. Both types of the cell have a liquid environment known as the cytoplasm. So this liquid thing in which a prokaryote cell, the DNA floats and the ribosomes present. So these things, the floating liquid environment, that is called the cytoplasm. And this cytoplasm present in the both the cases, either it can be a prokaryote cell or it can be the eukaryote cell. So these are the, now you can say these are the following similarities which can be present inside the both the cells. Now, if we see the cells are smaller and simpler in prokaryotes, so commonly known as bacteria. Most of the cells which come under the prokaryotes, they are the bacterial cells, and their size is like 10 to the 100 micron in size. So if a cell is larger than the 100 micron, then we cannot consider it as a prokaryote cell. They always the single cells, unicellular or filamentous. Filamentous in that they are strings of the single cell. At that time also when they look like a filament, okay? Still then, I will show you the diagram. This, I think I already showed you the filamentous type of diagram. Wait a minute, I will show you here. Yes, this is the filaments. Okay. Still, if they are filamentous, then sometimes they just look long, but they are having a single cell inside them. So these are the cell outlook of a prokaryote cell. There are, you can see the a microscopic diagram. This is the SEM diagram basically. This one. These are the prokaryote E. coli bacteria on the head of a steel pin. Okay, the steel pin. That is a very pointed little tiny thing, and you can see the thousands of the bacteria. Okay, E. coli bacteria. They are present on just one tip. So how? So now you can imagine how small they are in size, in the sizes. So now we will also see here. This prokaryotic cell are simply built. If we take an example of the E. coli cell here, so the capsule, slimy outer coating. So basically, why we are taking this here? So everything, whenever you go through the next topics, there will be a bacteria, viruses, or other microorganism. So you need to know the basic differences between a bacterial and virus cell. So here we are taking an example of a bacterial cell. So you can see the capsule, that is slimy outer coating. This is present on the outer surfaces on the wall. Why it is present here? Because the slimy outer coating, most of the infection or anything which is dangerous for the life of a bacterial cell, that can be captured in this slimy layer or this outer slimy layer. That's why this slimy coating have to be present. Cell wall tougher middle layer. After the slimy layer, we are having a cell wall. And cell wall is not slimy like capsule. It is a tough layer. Okay, so if something has come in the that slimy outer region, they cannot have to be penetrate inside the cell wall. After this cell wall, they are having the cell membrane. So basically, you can see how their defense mechanism work very appropriately for do not have any kind of damage or any kind of harmful effect inside the cell material. This cell membrane, that is a delicate inner skin, that cell membrane is basically present here just to cover that cytoplasm in the cell. There is no other major function that is done by this cell membrane. The just major function is there just to cover the cytoplasm. Now the cytoplasm, this is the inner liquid filling, okay? Anything which is present inside a cell that is basically floating inside this inner liquid filling. DNA is one big loop. You can see that this inner blue material, the whole blue material. This is the DNA that is spread out throughout the cytoplasm. So this DNA basically a big loop that present inside the cytoplasm. Pili, that is for sticking on the thing. This pili, you can see the light, small thread-like structure. So basically, they're for the sticking of the thing. If bacteria go for a surface, they need to be stick that particular substratum or surface. So with the help of these pili, they can be add up or they can be attached to a particular kind of surface. Next, the flagella. So flagella is the long thread here. Okay, so this long thread flagella, this is basically present here for the swimming or movement. So these all movement going one place to another place, that all directional movement can be done by the flagella movement. Last one here, inside the cell, that is a ribosome. They are the building block, they are the protein factories. So their major work here to perform is the protein synthesis. So whatever the code has been spread by the DNA or sent by the DNA, on the basis of their codes, these ribosomes, they make the protein for the cells. Now their lifestyle, how they live their life. So they can be unicellular, just a single cell can be present. They can be formed in the colony if they like some social life. They can be present in a colonical version, like the 50, 100, and 2000, 5000, the number that depend on the species. They can be present together. Filamentous. In this filamentous type of chain, they make a beaded chain. That like some beads are attached by one. So these our simple long thread, or you can say this filamentous chain, is particularly a arrangement in a linear direction. So at that time we call them the filamentous, in which there is a formation of a chain of a simple single cells. Now, if we see their prokaryote feeding process, okay, how they eat, how what by which they are getting food here. So first, very important thing here, the photosynthetic. So if they are the bacteria, and if the bacteria come under the living thing, so these photosynthetic way of bacteria getting food is energy from the sunlight. They made the food by, as the plant made the food by the photosynthesis process, same, they can make the food from the sunlight. Second one is a disease causing, free on living thing. Like we are having so many bacteria inside us, okay, and also that bacteria which cause disease in any body. So at the time of disease spreading, what they're doing there actually, they are just taking the nutrition to grow, and when someone will take our nutrition from us, then at that time we are feeling weakened, we are feeling like we are having the disease, but the same time bacteria are eating from us. So that is the way for them to eat and their survival, but that can cause several diseases inside our body, human body or animal body, even in the plant's body. So by the disease causing, they also get the nutrition. Third thing here is a decomposers. So decomposers basically feed on dead things. So like if something got dead inside the soil, there are several of bacteria present. When we compost a body, or some tiny little insect get dead, or anything that food we just left out to decompose, so that all the thing can be work as a food material for the small prokaryote cells. Now, eukaryote, if we talk about their shape and their structure, so eukaryotes are bigger and more complicated. So, how they are bigger and how they are more complicated? So, basically, they have lots of organelles inside them, so they are bigger. Because if there's something which do not have many things to store, that's why the size will be small. So, the prokaryote itself. But as we are talking, the eukaryotes have a lots of organelle to cover inside them. So at that time, these should be bigger in size. They have the chromosomes. multiple chromosomes, not a single chromosome like the prokaryote. They can be multicellular. So, because they are having a different kind of organism which led to the cell division and happening a great variety of the cell division, meristematic cell growth, differentiation, they can be the multicellular organisms, include animal and plant cell. They are having both the cell present inside the eukaryote. So they are having the animal cell, they are also having the plant cell. We cannot see this kind of differentiation in our simple prokaryote cell. They do not have that luxury to be converted into the animal cell and the plant cell. But as a eukaryote cell, it is a complex one. They have this fashion to a vein that they can be the animal cell and they can be the plant cell. Organelles in here, the membrane buds and parts, they are the mini organs that have unique structure and functions and located in the cytoplasm. That is very simple. They are having a large number of organelles inside their plant, inside their cell body, which are the membrane bound. Now we will see one by one the cell structure of a eukaryote. The very first is the cell structure is cell membrane. That is a delicate lipid and protein layer that, or you can say that it is the kind of the skin, as our body have a skin, the outer membrane. The same the cell membrane work for the eukaryote cell, and it is made up of the lipid and protein found in all the cells. This is a common feature for all the cells. Nucleus. If we talk about the nucleus, there are a very well membrane bound evolved nucleus having the chromosomes. Okay, so they are having a multiple chromosomes in them, as also they are having the spores. You can see in the diagram also. This is our nucleus, and this is a nucleolus. We will study it in next slide. And they are the ports, several ports basically for going in out. So they need some doors like we are having in our home, several doors. So same the ports are present in the nucleus membrane for going in and out. Now the nucleolus, this is very important location and basically location for the ribosome factories. So nucleosomes are the channel or the made RNA basically. So when the central dogma happened, like a DNA give the information to RNA, RNA give the information to the ribosomes, and they making proteins. That all thing is governed by this nucleolus. So nucleolus have a very major role to play inside a eukaryote cell. This is like a queen to the cell eukaryotic cells. Now the mitochondria. This is also very important, important part of this. If you can call them they are the vital one. Like mitochondria are basically the energy production source of cell. We can also call them the powerhouse. Okay, they are generating the entire ATP molecule by the various cycles happen inside the cell. So they are the units which make energy. So the more energy the cell need, the more mitochondria it has. So whatever the energy, whatever the demand that come through the cell, because every work, every function, what they need energy to do several kind of functions. So basically, mitochondria work on their need, and whatever the thing they desire, and whatever the energy they desire for their work, mitochondria provide them. Next, the ribosomes. As we already talking too much about the ribosomes, so they build protein from amino acids in cytoplasm, maybe free and floating, maybe attached to the ER. So the location, if we talk about the location of ribosomes, so they can be free floating inside the cytoplasm, freely float. They do not have to attach anybody, but sometimes they have to be attached to the part or organelle of the cell. At that time, that cell organelle have to be attached to the ER, endoplasmic reticulum. And when these ribosomes are attached to the ER, at that time, this ER is called rough endoplasmic reticulum because there are a layer of the ribosomes present on the ER, and Basically, the ER feeling like very having a rough surfaces. That's why we call them the rough endoplasmic reticulum. These ribosomes made of RNA. So basically, this the functional unit derived by the RNA unit. So whatever the proteins they will be developing, building, that is by the RNA molecule, which take their coding, which have the information from the DNA molecule. Now, the endoplasmic reticulum, they may be smooth if ribosomes are not present on their surfaces, so they can be the smooth one. But if the ribosomes are attached to them, they can be the rough one. So basically, they build lipids and carbohydrates. So whatever endoplasmic reticulum function for the buildment of lipid and carbohydrate, they also give a particular shape to the cell because they present at the both the ends of the nucleus. You can see in the diagram. Okay. So as a ear present here, they make a proper, give a proper shape to the cell. That's why they are very important. Next, the Golgi Fitting. You can see these tiny things at the corner. These are the Golgis. So takes insects of raw material from ER. So they are the delivering agent. This is a delivery boy. They take things from ER and take it out from the cell membrane. Or basically they are having the vesicles inside them which help them to the formation and transportation to from one place to another place in the cell. Now here we come with the lysosomes. So they are the sac filled with digestive enzymes. So basically all the digestion part in our body, the digestion work in our digestion system in our stomach area. So these are the lysosome which basically filled with the digestive enzyme instead of the cell. Basically they take up all the excretory material inside them and digest food absorbed by the cell. So sometimes they are also called the suicidal bats because they sometimes they just burst down and made the entire cell dead. So at that time they are also called the suicidal bat. They are not as sweet as they look. Now the centrioles, they are the pair of bundled tubes here you can see in the diagram. Basically, their function is at the time of cell division. Until and unless if the cell division is not happening in the cell, they do not perform any kind of major role. Now the whole cytoskeleton, this cytoskeleton, made up of microtubules, a small tubule-like structure which form this whole cytoskeleton of a eukaryote cell, found throughout the cytoplasm, not a particular shape, or basically they give the cell a shape. If we talk about like the ER, they also are responsible for the whole shape or the giving the shape to the cell map. Now, there are some structure that is found in the plant cell, additional structure, not in that are found in the eukaryotes of the animal cell. There are some special structure. The very first special structure here is the cell wall. This cell wall, this is not present inside the animal cell. Animal cell just have the cell membrane, but in the plant cell, they are having a very strong made up of cellulose for the protection and glue. These are the major characteristic of a cell wall because plant have to be very, plant have to go through very rough patches. So that's why they need something which protect them. So that's why the cell wall having a major role to play inside the plant cell wall. They are having a large vacuole. Animal cell, they do not have a very large vacuole, but you can see in the diagram. The nucleus is looking very tiny in front of the vacuole. So vacuole is a very major role occupying because this vacuole, basically what they do, they keep the cell pressurized or most of the time they are the working as a storage material. They can store water, they can store food. We take all our food from the plants. Okay, so now when the plant is making food, they just cannot make the food for their own process, they also made the food for us too. So at that time, the vacuoles have a very key function to store all the things inside them. So that's why the vacuole is present inside the cell membrane, because they have a very important role to play in the storage material. That's the chloroplast, and this is a very important feature of the plant cell. How? Because plants do the major function of the photosynthesis, hence they are called the autotrophs. So this autotroph feature, this whole autotroph journey is basically depend on these chloroplasts. These chloroplasts trap the solar energy and convert them into the food energy. So that's why this is all only present inside the plant cell. Now you can see there are the many differences between the animal cell and plant cell. Like I will summarize it for you. The very first difference is that they having the cell wall, and the plant animal cell they do not have cell wall. Secondly, the plant cell have a large vacuole. They do not have any large vacuole. Thirdly, they have the chloroplast in them. Animal cell do not have any chloroplast in them. They also have this rigid structure, or you can say sometime they are having the extra potential sap that is not present inside the animal cell. So these are the major difference, or you can say the three major difference: vacuole, chloroplast, and plant cell wall. These are the three major differences which is present between the animal cell and plant cell. Now you can also see how the structure vary. Cell membrane present in both, nucleus present in both, nucleolus present in both, ribosomes yes, ER yes, Golgi yes. Now centrioles, and that's sorry I forgot. Centrioles is not present into the plant cell, but they present into the animal cell. Plant cell at the time of division they do not need any kind of this central working. This is a key role of the cell synthesis pursued in the animal cell. Cell wall not present in animal cell but present in plant cell. Mitochondria present in both the cases because both need the both need the ATP's energies to do their functions. One big molecule animal cell do not need because they do not have to store that kind of amount, but plant cell really do. Chloroplast, animal cell, they do not have any function related to the chloroplast because they are not the autotrophs in it. But plant cell have the function because they do the photosynthesis. Now, last the cytoskeleton, you can see here. This is present both the cases, animal cell and the plant cell, because both the cell need a shape. That's why this present in both the cases. So they can be the multicellular cell, and because this multicellular feature of the eukaryote cell, they make them for suitable of a specialized job, because cell can work together as a tissue, and tissue can work together as a organ, and organ can work together as an organism, and this is the key dogma for all the process in the organism system. So now these are the basic some advantage of each kind of cell architecture. Prokaryote simple, easy to grow. Eukaryote can specialize. Prokaryote fast reproduction, multicellularity in the case of eukaryote, all the same can build large bodies. So these are architectural difference. I think we all go through about these. Just now, as you already studied a lot about the prokaryote, eukaryote, plant cell, animal cell, now I am sure that you can give the answer of these following questions. Like, what is the name of the region where double-stranded single circular DNA is present in a prokaryote cell? In prokaryote cell, ribosomes is the true domain of which prokaryote classify into these two means. Water sample from Earth. This is majorly a classification based question. Which of these are characteristic feature of the prokaryote cell? Already told you several special character of both the cell. Now you can answer those. Difference between eukaryote and prokaryote cell. This is all we already studied. In prokaryote, the hair-like outgrowth which attach to the surface of other bacterial cell are attachment. I told you no, that they have some thread-like structure for the attachment. These also. A component of prokaryote cell. What are the components? Process of recombination in prokaryote cell take place in way. What are the way? The flagella of the prokaryote and eukaryote cell vary in. So majorly flagella present sometime in both the cells. What are the key differences in? So these all the questions I think you might answer after all the lecture. Still, if you have any problem, you can just message me and ask me whatever the confusion is. These are the references for the very nice books and the paper, and even you can go through the online survey of that and some research digging, and might get all the answers you wanted. Okay, class. I think that's all for today. In next class, we will see the some tissue formation.